Let us see how new strand of DNA is synthesized. That is, what is the chemistry of synthesis of new DNA molecule. So two key substrates are required for the synthesis of DNA to proceed, and these two substrates are deoxynucleoside triphosphate, and the second one is primer template junction. So these two substances are necessary so that the DNA synthesis can be started. So in case of first requirement, which is deoxynucleoside triphosphate, so there are four types of deoxynucleoside triphosphates, and they are named as DGTP, DCTP, DATP and DTTP. These four DNTPs, these are required. So DNTPs means deoxynucleoside triphosphates. I hope you are aware of these deoxynucleoside triphosphates. These are the nucleotides which are present. So the nucleoside triphosphates have three phosphoryl groups attached to the 5' hydroxyl of the deoxyribose. Deoxyribose sugar is present, as you know, in DNA, and to the 5' carbon of this deoxyribose, three phosphate groups are attached. So that in this way it becomes nucleoside triphosphates. So in these three phosphates, the innermost phosphoryl group is called the alpha phosphate, while the second and third, that is the middle and outermost groups, are called beta and gamma phosphates respectively. So as you can see in this figure, this is the deoxyribose sugar, and it contains one base, and the base may be adenine, thymine, guanine or cytosine. Any one of these four bases may be present, which is attached to the 1' of this deoxyribose sugar. And here to the 5' of this sugar, three phosphate groups, 1, 2, 3. Three phosphate groups are attached. So in this way it becomes deoxyribonucleotide triphosphate, three phosphates. The innermost phosphate, or the first phosphate, you can see this is called alpha phosphate. The middle phosphate or the second phosphate group is called beta phosphate, and the outermost or third phosphate group is called gamma phosphate. So this is deoxyribonucleotide triphosphate. So this is the basic requirement for the synthesis of new DNA molecule. The second important substrate for DNA synthesis is the particular arrangement of single-stranded DNA, ssDNA, which is normally called ssDNA, and the double-stranded DNA, which is normally called dsDNA. So our dsDNA, double-stranded DNA, and a single-stranded DNA, they have a particular arrangement. So this is another requirement for the synthesis of new DNA molecule. This particular arrangement of single-stranded DNA and double-stranded DNA is called primer template junction. So you can see in this arrangement two components are involved. One is the template and the other one is primer, and the template and primer, they are joined together. That's why we say this is a particular arrangement of single-stranded and double-stranded DNA. So in this figure you can see this is the primer template junction. So in this this is the primer, and this strand of DNA is template. So where this primer will be attached to this DNA molecule. This is a specific primer template junction. So this is necessary for the synthesis of new DNA molecule.